Mitosis and meiosis. Mitosis. During interphase, the cell grows in shape, replicates its DNA, and prepares for mitosis. Chromatids are duplicated to form sister chromatids. During prophase, the first phase of mitosis, duplicated chromosomes condense and become visible. The centrioles move to opposite sides of nucleus and produce spindle fibers. Nuclear membrane disappears. During metaphase, the second phase of mitosis, chromosomes line up at the equator of the cell, and centrioles are located at the poles of the cell. Also, spindle fibers attach to kinetochores. During anaphase, the third phase of mitosis, the sister chromatids are pulled apart from their centromeres. In telophase, the final phase of mitosis, the centromeres spread out into a tangle of chromatin. A nuclear membrane reforms around each cluster of chromosomes. The spindle fibers break apart. Cytokinesis for animal cells, the cytoplasm is pinched into two equal parts by cleavage furrow formation. Each daughter cell has their own nucleus and organelles. In cytokinesis for plant cells, the cell divides by using a cell plate made of vesicles, helping it divide into two equal parts, since the cell wall is not capable of forming cleavage furrow.
Meiosis. Like mitosis, at the start of the division, the cell undergoes interphase where the chromosomes duplicate. DNA is synthesized, number of organelles increase, the cellular mass increases. At the end of interphase, chromosomes are formed as sister chromatids, which are genetically identical. During prophase 1, the sister chromatids that have been paired in the interphase connect to the homologous chromosomes. Homologous chromosomes can be described as a pair of chromosomes which have the same order of information with varying coding. The combined structure is called tetrad, and the process of pairing is called synapsis. After synapsis, tetrads exchange genetic material within each other, which is called crossing over. The chromosomes overlap each other to create a junction to exchange material, which is called chiasma. Spindle fiber forms, and nuclear envelope disintegrates. The tetrads are aligned at the equator of the cell. Spindle fibers then connect to the kinetochores of the chromosomes. However, the orientation of tetrads can differ, which affect the connection of spindle fibers compared to mitosis. There are three main principles, which are each chromosome is attached to one pole only, not both. The homologous chromosomes in a given tetrad should go to different poles. The orientation of tetrads is random, so every chromosome has the equal chance to attach to a certain pole. The process is very similar to mitosis, as the homologous chromosomes are separated. This separation of homologous chromosomes halves the total number of chromosomes, so the daughter cells becomes a haploid. Like mitosis, the divided chromosomes go to separate poles of the cell. Cytokinesis also occurs during this phase. No chromosome duplication occurs between telophase 1 and meiosis 2. Two haploid daughter cells are formed. Meiosis 2. Meiosis 2 is exactly the same as mitosis. The haploid daughter cells divide to form new daughter cells, which is equal to 4 cells in total. Spindle fiber starts to form, nuclear envelope disappears if produced, centrioles move to opposite poles. Sister chromatids are aligned at the metaphase plate. The centromeres of sister chromatids separate, and each individual chromosome goes to a separate pole. Nuclei forms at cell poles and cytokinesis occurs. 4 haploid daughter cells are produced at the end.